finish up the problems from the kind of polypeptide amino acid protein review, and then after that we'll start our chapter on lipids. So we'll spend probably the first half of the class doing some problems together, and then the second half with a short lecture. And then lab today we're just going to be doing a titration, so I imagine most people haven't done a titration before, so we'll talk about a little bit about what a titration is. And then we're doing kind of like a simpler version of an actual titration. Titration is the one, if you see in the other labs, they have the tall burettes and they're like sitting there watching and it's like, yeah, that's where you use phenolphthalein to use an indicator, and as soon as your solution turns pink, then you have to stop. And so that's why they get to like the very end and they're just all looking at their burettes doing that. But yeah, that's one way to do it. You can also use a pH meter, which is a lot more accurate, but indicators which change colors, like the cabbage juice we worked with before, is how that was done originally. All right, so this is the problem we left off at, which was, I thought, a good problem. It said, what type of interaction would occur between lysine and glutamic acid and neutral pH, right? So you're going to have your little chart with all of your amino acids and their structure and R groups on them. And then what are we thinking about? Well, the identity of the amino acids, that says something about its properties. It says neutral pH, which makes us think about the pH or pKa of the I groups or the side groups and how that affects their protonation state. All right, so I'll give you guys a second to think about that and work on it, and then we'll go over it. All right, so for this one, the identities of the amino acids, we have lysine and glutamic acid. Just based off the names, which ones do you think is going to be a low pKa-R group and what will be a high pKa-R group? Or just based off of one of the names of one of the amino acids? Glutamic acid is probably going to have a low pKa, right? It's probably going to be easy for it to get rid of its proton, right? So glutamic acid, I'm going to do this on another slide. Glutamic acid, let me see what it looks like because I don't remember it. 1, 2, 3 is we have, as always, our alpha carbon, our carbonyl carbon, our C terminus, and our amino terminus has 1, 2, 3 carbons. And then OH, and the pKa for this is, I can't even read that. Does it say 6.73? I'll look it up. 4 point what? 4.15. Oh yeah, I was looking at the wrong molecule. 4.15. So this is a pKa is equal to 4.15. Then we'll do lysine. And lysine is the long one with the amino group at the end. Where is that? 1, 2, 3, 4 carbons. So we have, again, the NH2, alpha carbon, and carbonyl carbon are there for this part as part of every amino acid. And we have 1, 2, 3, 4, and then in H2N at the end. And then the pKa for lysine is 10.67. 10.67. So I just drew them in some arbitrary protonation state right now. Because I was going to adjust the structure. Pardon? Yeah, because that's probably, yeah, it's the same thing. It's just I haven't drawn the charge on it. I'm guessing that they have the neutral pH charges already drawn on the side groups here. Yeah, so you're never going to protonate already protonated amine group. But if you change the pH of the solution, so for example, what they're giving you on the chart is the neutral ionic state or the charge state of each of the side groups. If I gave you the, I said it was a pH 11 or something like that, then you would have to adjust what the charge is shown there. So we'll find out what they give us is actually, different. Because I did the same thing for glutamic acid. Like they have the negative form in there. I just drew it right here to highlight this. Okay, so we have our two pKa's. If I was going to highlight the R groups, what are the R groups here? Or actually, first I'll highlight what's the alpha carbon. Yep, the one connected to NH, and that should be NH2, right? So this is the alpha carbon for both of them, right? And the alpha carbon is there for both of them. Where's the carbonyl carbon? Yep, the carboxylic acid carbon, which would be this one. So that is going to be our carbonyl carbon. Blue is our alpha carbon, and the alpha carbon has what bonded to it? The R group and a hydrogen, right? And it's the chiral center for the amino acid, so it determines what? Yep, which enantiomer it is, right? If it's D or L, which is determined by the position of the amino group or the N-terminus on the amino acid, right? So that N group, the N-terminus is going to be the one bonded to the alpha carbon as well. So then this is going to be the R group, which gives the amino acid its identity and determines what for the amino acid? Protonation states its properties, right? So the properties of the amino acid, whether it's polar, non-polar, or polar charged, is determined by the R group. All right, so that's the breakdown of how we look and draw these amino acids. Look at and draw these amino acids. Okay, so now let's draw, a pH 7 solution for both of these amino acids. So if the pKa for lysine side group or R group is 10.7, what is going to be the protonation state for the amino group at the end of the lysine there, right? So the pKa, and I'll usually draw the pKa right next to the R group like that, so you know that. Because on an amino acid, we have at minimum how many different pKa's for a single amino acid? 2, right? Because we have our N-terminus and our C-terminus, and both of those have an associated pKa value. So we have a minimum 2. So I'll draw the pKa values next to the associated acidic or basic group that's on the amino acid. All right, and so what are the two protonation states we're going through? Are going to be deciding between for lysine. All right, we're not going to worry about the C and N-terminus right now, but what are the, would the deprotonated form has a charge of what? For lysine. It's the nitrogen, right? For lysine, the pKa value, right, is related to this amine group on the R group, right? So that's a nitrogen that we're looking at the protonation state of. So depending on its pKa, it's either protonated or deprotonated, so it's either neutral or positive, right? The protonated form is positive and the deprotonated form is neutral, right? So we're going between NH positive or NH neutral. And this is the rest of the amino acid. And so the pKa value, how does it compare to the pH of the solution? pKa is greater than the pH, which means what? Which version are we going to favor? The protonated version, right? So pKa is greater, that means protonated version is what we favor there. Protonated is positive for the amine group, right? So I try to keep it in the same order as I did it on the board over here. Right, this is the protonated form, and this is deprotonated. Right, we see it's protonated because it has an extra hydrogen. deprotonated, that just means it has an extra hydrogen. Because why? Because in acids and bases, it's really just hydrogen nuclei that are moving around. So here, the protonated form for nitrogen is positive, the deprotonated form for nitrogen is neutral. So this is going to be H3, and then the charge is going to be plus on the side group. So this means the side group is protonated. All right, what about for glutamic acid now? All right, the pKa, pH is equal to 7, we know that. The pKa compares, and we're going between what two states for oxygen? Negative and neutral, right? The protonated version is that version, is it going to be neutral, right? That'll be neutral, and then we're going between that and the minus, the negative charge on the side group here, right? So what we're looking at is this part of the R group right here, right? That's the only thing we're analyzing right now when we're looking at how these amino acids interact. We're not worried about the C terminus or the N terminus yet, right? Because these could be part of a long polypeptide chain, and both of those would be bonded to other amino acids, so they would have no charge on them. How does the pKa compare to the pH? Less than the pH. So that means that it favors what form? Deprotonated, right? And for oxygen, that means that the side group is what? Negatively charged. Negatively charged. And that this one should be positively charged. So this, I'm going to erase the hydrogen there, and then I'm going to add a minus sign. Right, so that's how we look at the amino acids, and we determine the protonation states. And we could do this for any part of the amino acid. Right, so another way I could phrase the question is, here's a single amino acid in solution. What's the total charge on this amino acid if it has a charged side group? And you're accounting for the N and C terminus there. Right, so that means you just add up all the charges. There's 2 negative, 1 positive. It's a -1 charge on the amino acid, and so on. And so, again, I remember we talked about when the amino acids are in this form where they're neutral, but they still have charges on them. Does anyone remember what the name was for that? That was kind of a funny name. It started with a Z, and then there's a W. What is the ionic form where it's a neutral molecule with positive and negative charges on it? Zwitterion, yeah. So it's the Zwitterion. Z and then a W, right? So it's a Zwitterion. Right, so when it's in its Zwitterion form, that means it has both positive and negative charges, but the total charge on the molecule is neutral.
On these, though, there's only a positive charge on the lysine and only a negative charge on the glutamic acid.
Mm-hmm.
Versus, like, if there were 2 negatives on the glutamic acid.
Mm-hmm.
Is that what you're saying?
For the Zwitterion? Or
no,
these
sorry,
ones?
not for the
Oh, when I was looking at the
Zwitterion.
total
Just
charge?
the
Yeah. So for example, if really both of these, to get the total charge, you would need the charge on the C terminus, the N terminus, and the side group here, right? And these tend to have a Zwitter ionic form too, even though they have 3 potential charges on them, because there's a pH. It's in, so if you guys remember that, that was called the isoelectric point, whereas where the amino acid is in its Zwitter ionic form. Carly?
Does the charge of the group affect the name at all?
It affects the classification, but it doesn't affect the name. So you don't say that this is, always an arginine. It's not a positive arginine or another type of arginine. Yeah. The only thing that affects the name is the enantiomer, so the stereoisomer that the amino acid is, and then just the identity of the side group. And that's it. Right. So is it D-arginine or L-arginine, really? And for amino acids, almost all of them, and all naturally occurring ones, are going to be L, because remember, for for carbohydrates, it was D, right? It was D sugars for all naturally occurring carbohydrates. But for amino acids, it's the opposite. Everything is L, right? Except which one? Glycine, right? And so why is glycine not an L isomer? There's no chiral center for glycine because it has 2 hydrogens on it, right? It has 2 hydrogens, it's not a chiral center. Okay, so now we've done all the pre-work for this problem, right? So what was the question actually asking? Well, it said, what type of interaction would occur between lysine and glutamic acid at neutral pH? So how would you then, looking at these 2 molecules, now with their appropriate charged R groups, how would you say they interact with each other? They could hydrogen bond, yeah, that's a good answer. If I said what was the strongest interaction between them, that wouldn't be a good answer, but they do hydrogen bond. How else could they interact? When we were thinking of melting points for materials, what had the highest melting points? What kind of compounds? Ionic compounds, right? And so if we look at these, then we could have an ionic interaction. And why can we have an ionic interaction? Yep, because we have 2 full charges on each R group here. We have a positive charge, a full positive, and a full negative charge. So ionic interaction would be the strongest interaction between these 2 because we have full charges, right? Everything else, hydrogen bonding, dipole-dipole, London dispersion forces, those are all partial charges from either fluctuations in a dipole moment, which is a partial charge on 2 atoms, or a special partial charge, which is the hydrogen bond.
So like, only one of them have a charge that would not be.
Yeah, you wouldn't have an ionic interaction. You can have dipole ion interactions. We have never talked about that, so we're not doing it. But they can. The charge interactions can get somewhat complicated, but we're only looking if you have 2 charges. That's how they interact with each other. And so this affects how protein structure a lot, ionic interactions, because you have charged side groups that want to stay really close to each other. Okay, and so if you had a protein that had a bunch of lysines at neutral pH, and let's say this protein was an enzyme, and it was an active site, what kind of molecules do you think it would attract? What kind of property molecules? What kind of substrates? Right, so if you had an enzyme, right, looks like this, there's a bunch of lysines that are pointing into the, what part of the enzyme would this be? The active site. And they're all positively charged at neutral pH, right, because we just showed that. What kind of molecules do you think that, or substrates do you think this enzyme would bind? Negatively charged substrates, right? So the substrate molecules that would all fit into the enzyme would all most likely be negatively charged, because that would be the predominant interaction that would occupy that active site. Okay. Does that make sense? How do we feel about writing the protonation states of the amino acids? Thumbs up, thumbs to the side, thumbs to the side a little bit. Okay. All right, so let's do another one then. And I'll give you an arbitrary arbitrary pH, right? So let's do tryptophan. No. We'll do histidine. So we'll do histidine. Histidine at pH equal to 3.0. At 3.0. And what is the total charge on histidine? All right, so that's given in the on the table, all the pKa values for all the C terminus, the N terminus, and the side group for histidine. All right, so there's a structure of histidine. Okay, so how does the pKa, let's look at the carboxylic acid side first. How does the pKa compare to the pH of the solution? pKa is less than the pH, which means it's in the deprotonated state. And for oxygen, that means it's minus, right? So it'll be -1 there. Let's look at the N terminus now, right? So this, again, is the protein backbone, right? If we snap amino acids together, those are the bonds that make up the backbone of the protein. So the amino acid, or the N terminus, the pKa, how does that compare to the pH of the solution? Greater than the pH of the solution, which means it protonated. So that means how many nitrogens do we have on, how many hydrogens do we have on that nitrogen? The N terminus, it should be 3, right? 3, and it should be then positive or neutral, right? Because that's what we're going between for an amine, right? Positive, right? It has 3 nitrogens in the protonated state. For an amine, that means it's positively charged. Then we have our last one, which is on the side group. The pKa is equal to 6.01. And how does that compare to the pH of the solution? pKa is greater than the pH of the solution. So that means that what form do we favor? Oops, sorry. pKa is less than. No, pKa is greater than. Greater than the pH of the solution. So that means we favor which form? The protonated state. So how many hydrogens should be on this nitrogen? So let's think about it. Nitrogen is positively charged when it has how many bonds? right? So think about if you have four hydrogens on nitrogen, that's +1 charge, right? And how many bonds are involved there? 4, right? With 4 nitrogens, it's four bonds. Here, how many bonds do we have to this nitrogen? 3 already, right? So that's why in the neutral form, this would be deprotonated completely. There would be no hydrogens on it. We're counting the number of bonds here. There's 3 already. So in the protonated, positively charged form for a nitrogen, we would have only 1 extra hydrogen bonded to it. Right, again, because the charge, if we remember from Lewis structures, the charge is related to the valence electrons and the bonding electrons. How many bonds are there? And then how many lone pairs are there on the atom? And that's what gives you the charge. In general, for nitrogen, that usually means you have a hydrogen there or not. The neutral form here would have no hydrogens in this case.
So just, I'm confused because it's got 4 there, 2 parts to the double bond.
Mm-hmm.
What's that? So I'm like, how did they gain more electrons? I guess I'm confused. Or more protons.
Well, it didn't.
Not
It didn't
protons.
gain more electrons, right? It's just the lone pair that was previously on
Yes.
the nitrogen
Uh-huh.
is now involved in sharing the lone pair with the hydrogen, right? Because if we have a hydrogen here, this hydrogen has 1 valence electron,
Okay.
and then this nitrogen has another valence electron that come together to then form the covalent bond.
Okay.
Right? So the electrons are now being shared between the two, so that's why we usually subtract the number of bonds minus the number of lone pair
Okay.
electrons on there.
Okay.
Right, so it's the number of valence electrons. Let's see if I remember this. Formal charge equals number valence electrons, which for nitrogen would be 5, minus the bonds, number of bonds, plus lone pair electrons, right? So if we have 2 lone pair electrons and 3 bonds, which is what a neutral amine looks like, right? R, N, R prime, right? We have 3 bonds here, so that's 3 plus, nitrogen has 5 valence electrons, 5 minus 3 plus number of lone pair electrons, 2, that equals a formal charge of 0. Now when we 5 minus 4 bonds for, let's do something like R, H, just something funny like this. We know that that has a positive charge, right? This is double bond something. The number of bonds, how many bonds are attached to that? 4. And how many lone pairs? None. So the charge should be 1. You don't have to remember how to do this. This is just why we have a positive charge. The easy way to remember this is 4 bonds to nitrogen, positively charged. 3 bonds, neutral. 2 bonds, minus 1. It's just gonna go the other way, minus 1. Yep. Yeah, we're never gonna give you a negatively charged nitrogen. Okay, so that's positive then, right? Because in it's protonated, it still has a hydrogen. And then what's the total charge on this? Charge is equal to minus 1 plus 1 plus 1, right? Because we have a minus 1 charge here, a plus 1 charge here, and a plus 1 charge here. So we add them up to get the total charge, and that gives us plus 1. So would this be a Zwitter ion then? No, because it has a total charge on it, right? It's, usually, to be a Zwitter ion, you need the molecule to be neutral.
So it would be 0 if this is.
Yeah, it would be a zero. I'm never going to ask you about a Zwitter ion for anything that has a charged side group, because it's complicated to think about. If it's alanine, for example, alanine has no charge on the side group. It's just between these two. It'll be a Zwitter ion if the pH is somewhere between those pKa values. And we talked about that we talked about it pretty briefly when way back when we discussed amino acids. Let's see if I have the polypeptides here. Right, this is how we calculated the isoelectric point for this lecture. Was if we had an amino acid, you have two pKa values. The isoelectric point is the pH at which the amino acid is in its zwitterionic form, right? And you just add the two pKa values together, divide it by 2, and then you get the pH that you have the zwitterion. But if you have 3 different pKa's, it becomes a little bit more complicated.
So that would be like the one that you said that has no charge, and then there would be the.
Yeah, so like for this one, for example, this is alanine, right? So this is just the methyl group as the R group right here. The only 2 charges here are on the C terminus and the N terminus. And you just add those 2 together, pK1 plus pK2 divided by 2, that gives you the pH at which you have the zwitterion. Okay. Protein maybe. All right, so that's the total charge on this amino acid. Okay, any questions on this? Is it starting to make a little bit more sense?
So
Ali?
if something is favored protonated, if it's just like a nitrogen, you would automatically add 3 hydrogens if you can? Or
You don't automatically add 3 hydrogens, you just fill in the number of hydrogens to give it a positive charge.
oh, okay, gotcha.
Right, because in the neutral form, even let's say it has, a double bond, and this is the neutral form, R prime or something over there. To get this to be a protonated amine, you need to then add a hydrogen there, and that's going to be a positive charge.
Okay, that makes sense.
And so, for example, if you have, a nitrogen that has 2 double bonds to it, you wouldn't actually ever protonate this because this is already positively charged.
Okay,
It would be impossible to actually protonate. Just so you guys remember, that's not possible. Don't do that. Okay. All right. So this would probably be like a multiple choice type question on a quiz or an exam. The specific sequence of amino acids is what level of protein structure? Primary, right? So the same way this is like a sequence of amino acids is like a sequence of DNA and RNA bases. So primary structure would be the answer. This is the sequence of amino acids. Secondary structure is things like alpha helixes and beta sheets. And these are single motifs within a single peptide, right? That's how we would describe this. These are motifs within a single peptide. And what forces usually hold secondary structure together? Or are largely responsible for that? Yeah, it is forces between usually parts of the backbone of a protein with other parts of the backbone. It can be between side groups, but it's usually parts of the backbone. So for example, when you have an alpha helix, what you have usually is the N terminus of one amino acid. The R groups are kind of going up. C terminus is going down like that. And you're kind of going up a helix. And the carboxyl group from another one, these form hydrogen bonds between them. So that'll be another H that's bonded to another N terminus. And so this forms the helix. Usually you have right? So in the lab on Monday, they talked about preferred, or in the problem we talked about 2 problems ago, the preferred orientation of the R groups in an amino acid. And it was like you had, one R group going up this, then another nitrogen, another alpha, or let me do this. Carbonyl, here's the peptide, the alpha, R group going here, alpha carbon, R group going down, right? There was the trans versus the cis configurations of the amino acid, right? So this is the rest of the amino acid here. This would be called what? What is this right there? That bond? Peptide bond, right? This is the peptide bond. Or the, what other type of bond? What functional group is that? Amide, yep, this is the amide functional group, right? So the peptide bond is between the carbonyl carbon and the nitrogen. So the C terminus and N terminus of one amino acid. And so it has this configuration because, preferably because the R groups aren't opposite sides of each other. When you have this configuration, remember this has another thing that we didn't talk about a lot, slight double bond character, so rotation around this peptide bond isn't that favorable. So the carbonyl oxygen usually goes up, and the nitrogen hydrogen goes down, right? And these two can hydrogen bond to each other, because this is a hydrogen that's bonded to a nitrogen, so it has that special dipole here. And then if you imagine that this is a big old helix, right? And then we erase these right here, so this keeps going around, and then it comes back something like this. The carbonyl carbon will then be facing up here, and then the nitrogen that is bonded to the hydrogen will be going down, right? And so these will be all along the protein chain, and so these two can hydrogen bond with each other. And usually for an alpha helix, it's like 4 amino acids away where you kind of get this hydrogen bonding happening. This is just to illustrate that hydrogen bonding is an important force for holding secondary structure together.
And beta, how does it look for beta?
So a beta sheet this is kind of probably looks a little bit more like a beta sheet just because I didn't draw it that well. A beta sheet is going to be, there's going to be a turn, and then another turn. So here we'll have carbonyls, and then here we'll have the nitrogens going up from the backbone, right? And so that'll be all along both sides of these chains. And you can have other things that reinforce this too, like disulfide bonds, right? So if we're looking at cysteine, cysteine is the one that forms disulfide bonds. We have one problem that kind of deals with that. Those also can hold secondary structure together, but also tertiary structure. Claire?
So the alpha helix is like more three-dimensional, and the beta sheet is.
Yeah, that's exactly right. Yeah, so sheet, that's exactly right. A beta sheet, you think of a piece of paper, a sheet of paper, it's flat, right? It's two-dimensional. Alpha helix is going through three-dimensional space like that. There's still hydrogen bonds, right? These are just everything's lining up parallel. That's why we have these things at each side. These would be kind of usually whatever the motif is that's named off of that, and it's a turn. So this could be the beta turn is sometimes what it's called. This beta turn it's a disordered part of the protein, and then it goes back into the beta sheet. But they have to line up like each other. In alpha helix, it's nice because the backbone perfectly spirals, and so you get the nice order going up and down the protein backbone. And so that would be secondary structure. And then tertiary structure. Structure, that's going to be interactions between different parts of a polypeptide. I'm going to write the rest of that on the next page. Copy, paste, tertiary structure. Interaction between different parts / motifs within a polypeptide. Right, so that might be an alpha helix interacting with a beta sheet. You can have interaction between, they could be part of the same long amino acid polymer, and they're just interacting with each other. And then the last one is quaternary structure. And that's going to be what? What is quaternary structure? What do we remember from that? So everything we've discussed at this point is interactions and aspects within a single what? Polypeptide chain, right? So everything's been within a single polypeptide chain. So we've already talked about everything within a polypeptide chain. What's the only thing that we could go to expand even farther out? Yeah, if we go between polypeptide chains, right? And now you have two different polypeptide chains that we then interact with, right? Interactions between polypeptide chains. Right, so now if we have one chain that has, a specific sequence that has different motifs, and those motifs are interacting with each other, interacting with another polypeptide chain that has a specific sequence with different motifs that are interacting with each other. Right, so that's the level of kind of quaternary structure when we're taking a step back there. And this is mostly hydrophobic interactions that determine that. So basically non-polar interactions, London dispersion forces are largely what determine quaternary structure. All right, it's hard to think of a large globule protein where one whole side of the protein is polar, right? That much positive or negative charge in a single area would blow apart a protein, right? So it's mostly, you kind of have to mix in a lot of everything, so it's largely dispersion forces that drive these interactions. Okay. All right, so here we have an amino acid sequence. You're asked to write the amino acid sequence for the following polypeptide chain. And then another question you can ask is how many water molecules would you need to hydrolyze the following polypeptide? All right, so for this question, how many amino acids are in this polypeptide? 3. How do you know? Because it's 3 R groups, right? There's 3 amino acids because 3 R groups, right? So this is an R group, this is an R group, and this is an R group. Every R group gives a single amino acid. Okay, so each R group gives each amino acid. Let's do the 3-letter sequence for this sequence of amino acids. I'll number them. We have 1, 2, 3. What's the 3-letter code for the first amino acid? What is that amino acid? We have 2 carbons in between the alpha carbon and the carboxylic acid group of the R group, so that'll be, yep, it should be glutamic acid, right? Oh no, your glutamine, yeah, it has an H2 at the end. Or, yep, so it'll be glutamine. So this is glutamine, so it's an amide at the end, not a carboxylic acid. The next R group, what's this one? Arginine, this is my favorite one. That's arginine. And then the last one, what is that? This one, we have a CH3 group over there, so I believe that's methionine, right? Cysteine is just a thiol group at the end. We never talked about thiols, but a thiol group is SH, RSH. That's a thiol group. And so this has CH3 instead of a hydrogen there, so that is a methionine. Right, so to come up with a specific sequence, we go from N terminus to C terminus, and we just write the specific three-letter codes. So what's the three-letter code for glutamine? Who is saying it? It's probably right. GLN, right? GLN. I'll get you guys a better structure sheet for the exams because they're a little hard to read on this one. GLN, and then what about for arginine? ARG. And then what about for methionine? MET. All right, so that would be our three-letter sequence for these amino acids. You can also have the one-letter sequences too. Each has their benefit, right? If you have 500 peptide protein, then you probably don't want to use the three-letter sequence. You probably use the one-letter sequence for that. All right, so that's our sequence. And how many water molecules would we need to hydrolyze this polypeptide? Yeah, so you say 2. So we need 2 H2O molecules. And why is that? Yep, because we need 1 molecule per peptide bond to hydrolyze it, right? Because 1 molecule per peptide bond. And where are the peptide bonds in the structure? We have the carbonyls, right? These are the carbonyls, carbons. And then what's next to them? The NHs, right? And so this bond between them, those are the peptide bonds. So we need 2 water molecules because we have 2 peptide bonds, right? When we're making an amide or breaking apart an amide, we gain one side of the molecule is what? And what are the 2 sides of the molecule in an amide? Carboxylic acid and the amine side, right? So we add the OH to the carboxylic acid part, so the carbonyl, and then we add the H to the amine side. So that one was on. That one could show up on the next exam, right, because it's still relevant for proteins, but that was on the last exam for the hydrolysis of an amide, right? So that was the general organic chemistry. It's important for biochemistry because, well, we're made of organic stuff. And so how we would write that is if we had RC double bond N RH, right? This could be an arbitrary amide bond. We add water to it. And what do we usually need for this reaction to happen? We need a catalyst. So that could either be H3O or enzyme or something of that nature. And that's going to give us our C double bond O, OH, plus NHHR, an amine. Right, and 1 OH from the water goes here, and then 1 hydrogen, the leftover hydrogen, goes onto the amine there. So this is the organic chemistry reaction, but it's important for synthesizing proteins. Okay, am I good to go to the next slide? All right. Okay, so here we have a single polypeptide chain. So this will highlight some of the levels of structure you can have within one polypeptide chain, right? This single polypeptide chain, you see, has multiple alpha helices here, right? These are usually represented by these ribbons in a protein structure. So this is like a graphical representation of a protein. And they interact with each other through, hydrophobic interactions, maybe some hydrogen bonding and whatnot. Within the alpha helix, what was the primary force of that? Hydrogen bonding, right? So these dotted lines in the structure would represent hydrogen bonds within the alpha helix. That's what holds alpha helix together. And so if this is one polypeptide chain, quaternary structure would be what? Yep, if we had another one of these that then interacts with that. And I was saying to Vanessa, you can imagine if you had two of these, you pull them apart, right? You break the quaternary structure first and keep pulling. You'll start separating the alpha helixes from each other. You keep pulling, you unwind the alpha helix until eventually you're pulling on what level of structure? Primary, which is covalent bonds, right? You're pulling on covalent bonds, right? So as you pull proteins apart, you slowly break the levels of quaternary, tertiary, secondary, and primary. So this would be hydrogen bonds. In the dotted lines, this would represent secondary structure, right? Because this is a single motif within a polypeptide. Okay. Okay, so here's another one. Myoglobin consists of only one polypeptide chain. So this is a protein that only has one polypeptide chain. So what's the highest level of protein structure you can have for myoglobin? Secondary is a single alpha helix motif, right? So if you look at the previous example, let's pretend this is myoglobin, right? It's a single polypeptide chain. What's the highest level of structure you have here? Right? If this is a single chain, right? So we don't see any breaks in the chain anywhere. This means it's a single polypeptide chain. You see how many alpha helixes here? 4, right? And these alpha helixes are interacting with each other. But you don't have another peptide chain interacting. But that would be what level of structure? If another peptide chain was interacting with this peptide chain. Quaternary, right? Because it's 2 separate peptide chains. Here, within one peptide chain, we have 2 alpha helixes that are interacting with each other because the chain is so long that it folds on itself and they interact with each other. So what level of structure do you think that would be? Tertiary, right? So this would be tertiary because we have 2 elements of secondary structure interacting with each other. So using that, pretending it's myoglobin, we show that secondary structure is, not secondary, tertiary. Tertiary structure is the highest level of structure within a single polypeptide chain.
So, number 4, you said it was secondary because it's just interacting with the one helix, but
Yeah.
the dotted lines were in between those two
Yeah,
helixes.
that's exactly right.
Make it tertiary?
If you had 2 alpha helixes,
On the same
Uh-huh.
chain.
yep, and let's say that this was, so you know that it's part of the same chain, right? So there's no disconnects or anything here. These dotted lines would represent tertiary structure. And so the simplest polypeptides, the lowest level of structure they can have is primary structure, right? Because you can have a specific sequence and they don't really have any secondary motifs because they're not long enough to even form an alpha helix. but the largest structure you can have within a single peptide is long enough to have motifs and then long enough to have motifs interacting with other motifs. Good questions. All right, so we'll stop. Is kind of long, so if you guys want to try this on your own, label each of the amino acids, and then label the C and N terminus, and then all the alpha carbons as well. This is kind of what we did a little bit ago with the other molecule. So we'll end it there, and then this last question is, what kind of process is this? This is just kind of reminding us of other elements of chemistry and biochemistry. So here we have two cysteines, right? And cysteines can form what kind of interaction between parts of the chain? They form disulfide bonds, and it's saying, is this an oxidation or a reduction process, right? So we think about oxidation and reduction, what does that mean? Well, what do you guys think if you have this bond here forming? Do you think it's an oxidation or a reduction process? And how should we think about it? Right, we have hydrogens originally, right? And when we gain hydrogen, we follow the electron trend, right? So we gain hydrogen, it's like gaining electrons, that's reduction, right? So if we lose hydrogens, which is what we did here, we lost the two hydrogens on cysteine, and we form a sulfur-sulfur bond, the disulfide bond, then we've lost hydrogen. So it's an oxidation process. So that's how you would think about this process, as in the gain and loss of oxygen, or hydrogen. We lost H, oxidation. Why? Because gain O, oxidation, lose electrons, oxidation. Both of those don't apply here. It's easier to think about in terms of losing hydrogen. And we have the reverse for reduction. What's this?